I'm guessing that most of you are pretty familiar with the molecular structure of DNA. It's one of the iconic images of biology. The structure was solved originally back in 1953 by Watson and Crick in Cambridge using data obtained through X-ray crystallography by Rosalind Franklin. And so we've known about this for nearly 70 years now. And it's been incredibly influential because it was solving this structure which first enabled people to understand how DNA functioned as a repository of genetic information. And that in turn led to the whole modern science of molecular biology. So it's a hugely important structure to know about. But in science, we ought to do more where we can than just ask the what question. In other words, we want to know more than just what is the structure of DNA. We want to ask the why question. Why should these molecules fold up into this stable, highly intricate structure which is essential for its function? And that's what we're going to try and look at in this session.
And we're going to start by thinking about some fairly fundamental physics, so we're a long way removed for the moment from DNA. And just think about this situation. So you have to imagine that you've got two blocks, let's say, of metal, one of which is cold, one of which is hot, and we stick them so that they're sitting in direct contact with one another, and just ask the question, what's going to happen? And of course you all know what's going to happen, which is that heat's going to move from the hot block into the cold block, because that's what experience always tells you. And that's what you always find, so the heat will keep moving from there until eventually they're at the same temperature. And that principle is actually hugely important. It's sometimes stated in that form as the second law of thermodynamics, which is one of the fundamental laws that kind of explains how the universe works. We can think about the meaning of this a little bit more by thinking about what we mean by heat and by energy. So if I take this pen and lift it up into the air like this, then you know that I've given it a very specific form of energy, which is gravitational potential energy, and that if I let go of it, that form of energy will be transformed into another very specific form of energy. So there it goes, and you know that that energy has now become kinetic energy. But when it hit the ground, the question is, well, what happened to that energy then? And the answer is, well, it didn't stay as a very specific focused form of energy anymore. It's become dispersed. And it's become dispersed into various forms. We heard it hit the ground, so there was sound energy, which is vibrations in the air that happened. But of course those vibrations were triggered by the floor being made to vibrate when the pen hit it. So actually the energy's been dispersed through the floor, and the molecules that make up the materials in the floor are moving around, or were moving around. And now that energy is going to be spread widely out throughout the room. So that's really what we mean by heat. A quick way to summarize that would be to say that we gave it potential energy when we lifted it up. That changed into kinetic energy as it fell, and after it hit the floor, effectively the energy is dispersed as heat. So heat is disorganized energy. So if we think about that in the context of this, within the hot block, what we mean by saying it's hot is that the particles, the atoms in the metal, are all vibrating in random ways, spreading the heat energy out as widely as they can, whereas there's less vibration going on in the cold body. And what naturally happens is that as the molecules here vibrate, they tend to transfer energy to the molecules in that one, making them in turn vibrate, and as the molecules here on this side of the block vibrate, they will spread the heat out to that one as they sort of rub against one another and spread the energy out. And that's what always naturally happens. You can't imagine that it would ever happen that as all these molecules randomly vibrate, that it would force the heat to concentrate in one particular place. Common sense just tells you that the energy doesn't behave like that, that it will tend to become more spread out because of the random nature of the process. And that's the most fundamental way of thinking about the second law of thermodynamics. It's that when you consider the universe as a whole, everything that happens, absolutely everything that happens, always leads to an increase in the dispersal of energy. And the second law says that has to be the case in any process in which the universe as a whole has its energy becoming more ordered is just not physically possible. And it's just a statistical thing. It's just because of the random nature of energy transfer processes that statistics say it's impossibly unlikely for energy to become more ordered rather than less ordered. So that's the second law of thermodynamics. And what we want to do is think about the implications of that in terms of the DNA double helix. If this is our double helix, we've got two molecules. So the red one and the blue one represent two separate, very long molecules which are coiled around one another to make the double helical structure. And the question we have to ask is, what if it didn't do that? What if they weren't wrapped around one another to make this structure? Well, what you would then have, of course, would be two completely separate molecules. And if they're not locked into a specific structure, then presumably they could have all sorts of possible structures where they have flexibility. And so what we need to do is to think about what the implications of the second law of thermodynamics are for the folding up of these two separate chains into this very specific structure. And you can probably guess what the answer to that is, but let's just look at it in a bit more detail.
So this is an animation of what chemists call a random coil, which means that it's a long polymeric molecule like a DNA molecule, but with no reason to have a preference for a particular structure. And you can see that what happens is that it just keeps sampling all kinds of different arrangements, and because it's able to have rotation around the individual bonds along its backbone, it's able to change its overall shape as a function of time. Now, if we think about that in energy terms, all of those rotational motions around the individual bonds are a form of energy. And so what we're seeing then is that this has a very highly dispersed energy because all of these individual bonds are independently managing to rotate. Now you can imagine that if two of these molecules come together to form a double helix, then that dispersal of energy is much less for two reasons. First of all, because the two chains are now going to be locked together into the same place, so they can't spread their energy into separate parts of the universe because they're locked together into the same structure. But also, that freedom of rotation is going to be much less too, because although there'll be some freedom to move around within the double helical structure, you can see that it's likely to be much less because the forces holding the helix together are likely to make most of those rotations impossible. And while there'll still be some vibrational motion, there'll be much less dispersal of energy. So looked at from that point of view, the folding into a double helix looks like a statistically unlikely thing to happen. And we have to think about, so why does it happen?
Now you can ask that question about not just the folding of DNA, but about any process which leads to formation of a ordered structure. So you can actually ask it about the whole of biology. How is it possible that something as highly organized as a living system can exist? Is it breaking that second law of thermodynamics? But of course you can also ask it about simpler things like a crystal. A crystal is a highly organized arrangement of atoms. And the question is, well, shouldn't the second law of thermodynamics mean that crystals should always fly apart? And the favourable thing would be to have the molecules dispersed in a solution or a liquid state or even more so a gaseous state. So there must be something else going on. And what else is going on is to think about, well, when things become organised, it involves the formation of bonds. So in the context of the double helix, there must be bonds, attractive forces, in other words, holding the structure together. And when you form a bond, it gives out heat, because you can think of a bond as just something formed in response to an attractive force. So again, if we think about dropping the pen and think about why does the pen drop to the earth, the answer is because there's an attractive force dragging it in that direction, in this case gravity. And when it moves in the direction of that attractive force, it loses its potential energy, and in the end, as we discussed before, that potential energy is then dispersed as heat. So it's the same when a chemical bond is formed, that when the particles are separated from one another, they have potential energy, because when they come together, they're moving in the direction of an attractive force, and so they end up with a lower potential energy, and that energy ends up being dispersed as heat. So the justification for the existence of an ordered structure like a double helix has to be that the formation of the bonds that hold this structure together gives out energy in the form of heat. And heat, as we know, is a highly dispersed form of energy. So when we say it gives out heat, what we really mean is that all the molecules and other particles in the surroundings get more energy, which they spread out in a random way amongst themselves. So if we don't just focus on the double helix, but we focus on the whole universe, even though folding of the DNA into the helix makes the DNA molecules more ordered and therefore the energy they carry with them less dispersed, the universe as a whole becomes more disordered because of the heat that's given out. And that's what must be true for every event which leads to a more organized structure. So the whole of life, for example, depends on this principle that yes, you can make organized things, like DNA molecules or rabbits or whatever, but only at the expense of making the wider universe more disordered because of the heat given out by the formation of the bonds. That's a hugely fundamental principle. But it's not quite as simple as that to think about, because we're not talking about the folding of a DNA molecule in a vacuum. If we were, then it would be quite simple, because we'd just say, well, if it wasn't folded up, there would be essentially no bonds between different parts of the chain. Obviously there are bonds holding the chain together as a chain, but there would be no bonds at all between different parts of the chain, and we'd just form those bonds, so we would be comparing some bonds with no bonds. But it doesn't fold up in a vacuum, it folds up in aqueous solution inside cells. So what we actually have to do is think of this as an equilibrium in aqueous solution. So we've got aqueous DNA folded up into a double helix, and we've got to think about the aqueous unfolded state where there is no double helix. And we can think about that as a chemical equilibrium. So what we're really asking is what forces the chemical equilibrium in that direction so that the double helix is stable. Now it's worth introducing a word here, which is what we're talking about in this whole discussion, and that word is entropy, which is a concept you may have come across before, but effectively that's what we've been talking about, because entropy is a physical quantity which in effect measures the dispersal of energy in a system. So what we're saying is that it would appear that this is a low entropy system because the energy is highly organized, and this is a high entropy system. So the question we're really asking is what else is going on, given that the second law would say this ought to be the direction things are moving in, but we're saying, yeah, but it's not, it's moving that way. And so we've got to say, well, what else is going on which allows us to form this state, which in itself may have a lower entropy, but which leads to the universe as a whole having a higher entropy. Now you can ask that question about not just the folding of DNA, but about any process which leads to formation of a ordered structure. So you can actually ask it about the whole of biology. How is it possible that something as highly organized as a living system can exist? Is it breaking that second law of thermodynamics? But of course you can also ask it about simpler things like a crystal. A crystal is a highly organized arrangement of atoms. And the question is, well, shouldn't the second law of thermodynamics mean that crystals should always fly apart? And the favourable thing would be to have the molecules dispersed in a solution or a liquid state, or even more so a gaseous state. So there must be something else going on. And what else is going on is to think about, well, when things become organised, it involves the formation of bonds. So in the context of the double helix, there must be bonds, attractive forces, in other words, holding the structure together. And when you form a bond, it gives out heat, because you can think of a bond as just something formed in response to an attractive force. So again, if we think about dropping the pen and think about why does the pen drop to the earth, the answer is because there's an attractive force dragging it in that direction, in this case gravity. And when it moves in the direction of that attractive force, it loses its potential energy, and in the end, as we discussed before, that potential energy is then dispersed as heat. So it's the same when a chemical bond is formed, that when the particles are separated from one another, they have potential energy, because when they come together, they're moving in the direction of an attractive force, and so they end up with a lower potential energy, and that energy ends up being dispersed as heat. So the justification for the existence of an ordered structure like a double helix has to be that the formation of the bonds that hold this structure together gives out energy in the form of heat. And heat, as we know, is a highly dispersed form of energy. So when we say it gives out heat, what we really mean is that all the molecules and other particles in the surroundings get more energy, which they spread out in a random way amongst themselves. So if we don't just focus on the double helix, but we focus on the whole universe, even though folding of the DNA into the helix makes the DNA molecules more ordered and therefore the energy they carry with them less dispersed, the universe as a whole becomes more disordered because of the heat that's given out. And that's what must be true for every event which leads to a more organized structure. So the whole of life, for example, depends on this principle that yes, you can make organized things, like DNA molecules or rabbits or whatever, but only at the expense of making the wider universe more disordered because of the heat given out by the formation of the bonds. That's a hugely fundamental principle. But it's not quite as simple as that to think about, because we're not talking about the folding of a DNA molecule in a vacuum. If we were, then it would be quite simple, because we'd just say, well, if it wasn't folded up, there would be essentially no bonds between different parts of the chain. Obviously there are bonds holding the chain together as a chain, but there would be no bonds at all between different parts of the chain, and we'd just form those bonds, so we would be comparing some bonds with no bonds. But it doesn't fold up in a vacuum, it folds up in aqueous solution inside cells. So what we actually have to do is think of this as an equilibrium in aqueous solution. So we've got aqueous DNA folded up into a double helix, and we've got to think about the aqueous unfolded state where there is no double helix. And we can think about that as a chemical equilibrium. So what we're really asking is what forces the chemical equilibrium in that direction so that the double helix is stable.
Okay, so in order to answer that question, we need to look in a bit more detail at what the actual chemical structure of DNA is like. So this is a particularly short section of DNA. It just has 4 repeats in it, and you can see, for example, if we look at these rings, that there are 4 of those. In biology, DNA molecules can be enormous. They can be billions of repeat units long, so this is just a very short example, but it lets us see what kind of molecular structures we've got which are going to be important potentially in folding up the double helix. So each of these repeats basically has three parts to it. First of all, there's this, which is a phosphate group, and that forms the link between each repeat unit. So we have to think a little bit about those and what their implications of having those is going to be for the folding up. Secondly, we've got this group, which is actually derived from a sugar called deoxyribose. And then thirdly, you've got this bit, and this bit is different in each of these four repeats because that's the way DNA works in biological systems, that there are four of these possible chemical structures that can go in this part of the repeat unit. And they're called the bases of DNA, and that's just because chemically they can act as bases in the sense that they can react with acids. But they too will have a very important role, as we'll see, to play in organising the double helix. So thinking about the entropy, you can see that it's along this backbone here that you've got lots of single bonds. So you've got carbon to oxygen, to phosphorus to oxygen to carbon. And all of those single bonds have this freedom to rotate. And that's what allows the unfolded state to sample so many different possible shapes and structures. And that's what gives it its high entropy. So it's really the locking of the angles of rotation in the backbone, which is the problem in entropy terms, which is going to have to be overcome for the double helix to be stable by formation of favourable bonding interactions.
So let's think first about these phosphate groups here, because the way they're drawn here is in a way a little bit misleading, because the point about that is that it's an acidic group. So that hydrogen, which is drawn there in the structure, can actually leave in the form of H+, so that you can actually get O-, and then the H+ can wander off into the solution. And the same here. And whether that happens depends on the pH of the solution that the DNA is dissolved in. But of course in biology that pH is very tightly controlled, and in cells it's not very far from neutral. And around that pH these will be fully ionized. In other words, they will all have lost their H+ ions. So very importantly, what that means is that each of these phosphate groups has a negative charge. So first thing I want you to pause the video and think about is, what are the implications of having those negatively charged phosphate groups on the chain for the folding up into the double helix? So remember, we're thinking about this process of a random chain like this becoming organised into a double helix like that. So what I want you to do is think about where you would expect the phosphates to end up on the chain. Would you expect them to be on the inside or the outside? And secondly, what effect would you expect having that array of regularly spaced negative charges? What would you expect the effect of that to be on the stability of this relative to an unfolded state? So pause and think about that for a few minutes, and then we'll talk about it and see where that gets us in terms of understanding this folding equilibrium.
So here's the double helical structure again, and those phosphate groups are these red and orange groups that you can see here. So the orange atom in the centre is the phosphorus, and the reds attached to it are those oxygen atoms sticking out into the solution. And you can see that exactly as we predicted, those negatively charged oxygens are sticking out into the solution. And that's really important, because that means that at least they're not being dragged away from the water, and that would be a very difficult thing to compensate for. It would be a very unfavourable thing. So that's not something which is helping to make the structure stable, but it is something which is helping to define what the structure is, because alternative possible structures which drag those phosphate groups away from the water would be less favourable. And so that's a crucial thing here, because we're not just asking why does it fold at all, but we're asking why does it fold into this particular structure, and having the phosphates on the outside is very important for that. Having said that, though, there is still an issue, which again you can see quite clearly on here, or possibly even more clearly if we change it to a space filling representation like this. So here you can see the phosphate groups clearly. So there's one, and there's the next one in the chain, and there's the next one in the chain. And you can see that the effect of folding up into the double helix is that you bring this phosphate group into quite close proximity with that one. Okay, so these are in adjacent turns of the chain. And that's true all the way around, this one and this one, this one and this one. And of course, what that is doing is increasing repulsion between those negative charges compared to what you would get if the chain was unstructured. So actually the formation of the double helix is unfavourable in terms of repulsion between the phosphate groups. And you can demonstrate experimentally that that's true, because if you add a higher concentration of salt to a solution of DNA, you find that the stability of the double helix increases. And the explanation for that is that you can get positive ions from the salt to sit in these spaces in between the phosphate groups. And obviously by having a positive ion there, it makes it much less unfavourable to have the repulsion between the two negative charges here, because it's compensated by having a positive charge there. So far, we haven't got anything helpful in terms of understanding why the double helix should be stable. We've seen what you can do to minimize the instability by keeping the phosphate groups on the inside, but there's not been anything positive in terms of why does it do this. So the next thing we're going to look at are the bases. So let's have a look at the chemical structure again and see what the sort of personality of these bases are. Remember the bases of these groups here and here and here and here. And the thing that probably strikes you immediately is that they have lots of nitrogens and oxygens in them. And also, more specifically, some of those nitrogens are in the form of NHs, so they have nitrogens and hydrogens attached to one another. And again, if you've studied a bit of chemistry, you'll probably immediately recognize what kind of intermolecular forces these bases are going to be very good at. So again, what I would like you to do is pause the video for a moment and think about what's going to happen when the double helix folds up in terms of the interactions formed by those bases. And in particular, think about the fact that this is all happening in aqueous solution and what the implications of that might be. Well, what you may have realized is that these bases are going to be astoundingly good at forming hydrogen bonds. So let's just remind you what a hydrogen bond is, and then we can see how those play into that structure.
If you have an OH or an NH or an FH covalent bond in a molecule, there's a real mismatch in terms of how strongly these nuclei attract the shared electron pair. So these three elements are very electronegative, which means that they attract the shared pair much more strongly than the hydrogen does. And so in each case, this atom becomes significantly negatively charged, which we write as delta negative, and the hydrogen becomes significantly positively charged. And when that happens to hydrogen, because it has no other electron pair to shield the nucleus, if you pull this electron pair strongly enough this way, that becomes very attractive to other atoms, because that partial positive charge is very focused on its tiny nucleus in the middle, not shielded by any other inner electrons. And that's the key to hydrogen bonding. So in water, for example, water molecules stick to each other by forming hydrogen bonds kind of like this, where you have an attraction between the delta positive hydrogen of one molecule and the delta negative oxygen of another one, and it's more or less just an electrostatic interaction. So the positive charge here is attracted to the negative charge there, and in particular it tends to point at one of the lone pairs on the oxygen, because that's where the sort of partial negative charge is most focused. So when you're thinking about hydrogen bonding, what you have to do is look for these kind of covalent bonds in the structures of the molecules that you're dealing with. So oxygens or nitrogens, or fluorine's not really going to arise because that happens in HF, hydrogen fluoride, but that's really the only place, so we can sort of forget about that one. So you're looking for OHs and NHs in the molecules, and you're recognizing that where those happen, you have the possibility for hydrogen bonds to form. The other thing about hydrogen bonds is that they're very directional, because if you think about this delta negative oxygen, it's going to be attracted to this delta positive hydrogen, but it's going to be repelled by the delta negative oxygen. So it's not just that it wants to get close to the delta positive hydrogen, it wants to get as far away as possible from that delta negative oxygen. This is something we don't really emphasize at A level, but actually when it comes to understanding structures, especially biological structures, it's really important. So a hydrogen bond wouldn't look like this, for example, and you would get away with a picture like that in an A-level exam, but it wouldn't really look like that. It's very directional. So what you've got to look for when you're trying to identify where hydrogen bonds might form is this linear negative, positive, negative, all in a straight line with the hydrogen in the middle. And basically the number of hydrogen bonds that an atom can form, hydrogen can only ever form one, oxygen can form two, and you can think of that as being because it has two lone pairs. So actually each of those can form a hydrogen bond. When you have nitrogen, it's generally forming three covalent bonds, as in ammonia, for example, and therefore it has only one lone pair of electrons, so it will just form one hydrogen bond, like that. Okay, so with those ideas about hydrogen bonds, what I want you to do is to now have a look at the picture which I included as an attachment to the email that gave you the link to this. And what that picture has is the structures of the four bases that make up DNA.
There are four flavours of base that are used in the construction of DNA, which are cytosine, thymine, guanine and adenine, which will abbreviate as C, T, G and A, and you can see them here in the picture. And what I've done is to provide you with two views of each one. So, for example, this is cytosine viewed from one side, and this is cytosine viewed from the other side. And the reason for that is that this black blob here is the point of attachment to the sugar, in other words, to the rest of the DNA molecule. And the bond between that point and the base is just a single bond, so you can rotate freely around there, and therefore it would be entirely possible for this base to flip over from this orientation to this orientation. And so when we're looking for hydrogen bonding possibilities, we need to be able to view the base from either side like that. So in these diagrams, what I've done is to show the the groups that could potentially be involved in hydrogen bonding. And I've color-coded it so that what we call the hydrogen bond donors, in other words, the atoms that could be involved in providing the hydrogens to be involved in hydrogen bonds, are shown, so that the NHs that could be involved are all illustrated. But the hydrogen bond acceptors, which are the nitrogens and oxygens, the partially negatively charged atoms, which can interact with the NHs, are coded in red. So if we just take one example of this, we can see how you can attempt to match these up and see what the hydrogen bonding possibilities are. So if we, for example, take a thymine, okay, we'll move this down to here, then we can look for things that can potentially form a complementary set of hydrogen bonds. So for example, if we take this cytosine, we can move it like this, and we can just play around. So you can see here we've created the possibility of just one hydrogen bond there between the NH here and the oxygen there. But that's all. None of the others come close enough to form hydrogen bonds. But if we slide this down a bit, then now you can see a possibility of two hydrogen bonds, because you've got NHN there, all in a line. Remember, the hydrogen bonds have to be linear like that. So that one's forming a hydrogen bond, but you've got OHN there as well. So you've got two hydrogen bonds forming potentially between a T and a C. And that's what I want you to do, really, is to either on a printout of this, to mark on the possible hydrogen bonds that can be formed, and then cut out the individual structures and try matching them up and see what combinations you can find that make at least two hydrogen bonds between bases. Because we're going to speculate that obviously you could always find one hydrogen bond, but we're going to speculate that two might be the minimum necessary to form some kind of structure. So just have a go at exploring that and see what combinations you can find that seem to work potentially as hydrogen bonding pairs. Now, you may already know what happens in natural DNA, and obviously you can confirm that those base pairs work, but don't consider only those, because what we're exploring here is why the DNA structure has the arrangement that it does. And so we shouldn't just explore the way it actually is. We need to understand why it isn't some other way. So have a go at that now, and then we'll explore those results a bit further. Okay, so let's explore that then, if you've had a chance to think about that a little bit. So let's take adenine as an example. So here is adenine. Let's move that down here. And then we'll look at which of the bases could possibly hydrogen bond to adenine. So actually there are two directions we could possibly form a pair of hydrogen bonds. So one is here and here, because you can see that we've got an NH being donated that way, and then we have a nitrogen here which could potentially be involved as a hydrogen bond acceptor. Or we've got this interface down here where we've got an NH there and a nitrogen there. And we're just going to focus on this interface just for this exercise. So let's then look at the possibility of adenine pairing to each of the bases in turn. So let's start with guanine. And you can see, if we slide guanine down to here, that we can quite easily form two hydrogen bonds between a guanine and an adenine. So you have OHN there in a line and NHN there. So we can make a 2 hydrogen bond combination between those two bases. If we look at possibility for thymine, we can bring that down here, and again you can see very easily that we can form 2 hydrogen bonds there between an A and a T in a similar kind of array. If we look at cytosine, it's not quite as easy, because if we bring it down here, you can see that the best we can really do is form 1 hydrogen bond like that, until we realize that actually, of course, the cytosine could be attached up here rather than down there. So in other words, it could be upside down relative to this drawing. So if we just flip it over, which we can do like that, now you can see that actually cytosine 2 could form a pair of hydrogen bonds like that if we change the orientation suitably. So all 3 of those base pairs are possible. Cytosine or thymine or guanine could all base pair with adenine. And indeed, we could ask whether it could hydrogen bond with itself. So if we take another adenine, the question is, can we form a base pair there? And again, it doesn't work like this, but if we flip it over so that it's approaching from the other direction, then you could see again that we can make a pair of hydrogen bonds there, NHN and NHN there. So actually, adenine could base pair with itself, forming a pair of hydrogen bonds. So what this exercise shows is that actually there are lots of ways of forming combinations of bases which can form a pair of hydrogen bonds between them. There is actually only one combination which can form 3 hydrogen bonds between the bases, and that one is G and C. So we'll just have a look at that one. So if we bring G down here, which is guanine, and then we bring cytosine in as well, you can see that in that case we can actually make an array of 3 hydrogen bonds. So we have NHO there, NHN there, and OHN there. So a CG base pair is a little bit special in the sense that it can have 3 hydrogen bonds rather than just 2, and that's the only combination that can do that. What I mainly want you to take away from this exercise, though, is that there are lots of combinations of bases which can form 2 hydrogen bonds between them, and therefore potentially sort of pair up through hydrogen bonding interaction. So let's have a look at how that actually works in the DNA structure. So here's our double helix structure again. So remember that the backbone made of the phosphate groups, which you can see because they stick out as orange there, and these 5-membered sugar rings there and there and there, for example, that winds around the outside of the double helix. But the bases actually form the core of the helix, and you can see them because you can see all the blue coming from the many nitrogens in the bases, and they're right the way through the middle of the helix. And if we just zoom in a little bit on that, like this, and then twist it round a little bit, what you can see is that the bases are indeed paired through hydrogen bonding in the way that we've been exploring. So here, for example, is an adenine, an A, and a thymine down here, which is a T, and you can see a pair of hydrogen bonds there. So you can't see the hydrogens shown in this structure, because when you solve structures of molecules by X-ray crystallography, which is how this was done, the hydrogens don't show up and you just have to infer where those are. So actually the hydrogen bonds here are shown between nitrogen and oxygen, and there would be a hydrogen in the middle there. But you can see the pair of hydrogen bonds between A and T there. And here, if we just rotate this a little bit more, here we've got a guanine and a cytosine. So we've got a GC base pair there, forming, as we said, the combination of 3 hydrogen bonds. And what we find is that those are the only base pairs you ever find in normal DNA. So you get A paired with T through a combination of 2 hydrogen bonds, and G paired with C through a combination of 3 hydrogen bonds. And if you studied DNA in the past, you know that that complementarity, G with C and A with T, is the key to everything. That's how genetic information is passed on. As these molecules make copies of one another, you always get that specific pairing. But the question we have to ask now is, well, why? Because while it's true that G and C was the only combination that could make 3 hydrogen bonds, we saw that A and T is by no means the only combination that can form 2. So we need to look in a bit more detail about why those specific combinations are used in forming the double helical structure, whereas others are not. So let's come back to looking at the bases at this kind of level and see if we can see what's particularly special about those combinations. So if we take our cytosine and move it down to here, and then we'll bring in the guanine that we saw pairs with it, and we can create that set of 3 hydrogen bonds there. Okay, so there's our CG base pair, NHO, NHN, OHN. Those are our 3 hydrogen bonds there. And then we can create the AT base pair, which was the one that we saw in the molecule, was the other combination that was formed. And if we want this one, don't we? So if we move adenine down here, we can form that pair of hydrogen bonds there, which you can see. So those are the 2 base pairs that we get naturally. So let's have a look now at what happens if we superimpose these 2 base pairs one above the other. So if we just drag this one across here and put it on top of that one, then you can see that they superimpose almost exactly. So those 2 base pairs have exactly the same shape and orientation, and that's very important. If we contrast that with one of the other base pair combinations that we made, we'll see that that doesn't always work. So, for example, we could pair T with another T, and if we drag the T down to here, we can form a pair of hydrogen bonds like that between 2 T bases. But if we try and then superimpose that base pair on the GC one and see what happens, then you can see that it's not now the same at all, because obviously the 2 bases are smaller. The 2 T's are both single ring bases, whereas G is a double ring base, and so that combination just has a greater overall width than the TT combination, and so they don't coincide at all, but also the angle is different, as you can see when we try and superimpose them at that end. That you can see that it's not just that this is closer in, this point of attachment here, but it's also not aligned because of the different base pair combination that we've got there. The same would obviously be true if we tried to pair two of the double ring bases, the purine bases, that then again it wouldn't have the same shape because they would be further apart. What about the possibility of making a non-standard combination of a single ring and a double ring? Let's see if that works any better. So if we move this T back up to the top, and let's see if we can make a TG base pair. So if we select this and then copy it, we can bring the G down to here, and you can see that we can make a pair of hydrogen bonds like that. So GT is capable of forming a pair of hydrogen bonds, NHO, OHN, like that. But again, if we now look to superimpose those on our original CG base pair, then you can see that although it now has the right sort of overall size, the orientation is different. So the 2 points of attachment are out of alignment. And what all of that suggests is that it's that reproducibility of the overall shape that's the crucial thing. And you can see how that makes sense in the context of the double helix structure. So the striking thing about this structure is just how regular it is. So you can see the helix just reproduces in an entirely consistent way all along this axis. You can see the base pairs are evenly spaced through the structure, and if we look at it end on, just by rotating it round like this, you can see very clearly that it has this very regular reproducible cross section with the bases located in the middle like that. And it seems likely then that the requirement for G to pair with C and A to pair with T is linked to the fact that you couldn't form as regular structure as that if the points of attachment were distributed differently each time you made a base pair combination. You can see that you would have to distort the backbone in order to bring those points of attachment into alignment if you had other combinations of bases. So what we're saying is that it's not so much just the complementarity of the hydrogen bonds that matters, but actually it's the regularity of the helix structure that's important in determining what base pairs will be formed. And we can get a little bit more insight into that by looking at a space-filling version of this model. So when you draw molecular structures out like this, it's kind of misleading because the atoms have no volume, and it just shows a sort of map of where the center of each atom is. But if we put realistic relative sizes on the atoms, we can get a slightly more realistic picture. So actually we can do this very easily here just by choosing that. So that's now what's called a space-filling model of the double helix. And again, you can see the backbone around the outside with the phosphate groups and the sugars in between, and then you can see the base pairs in the middle. And what I really want you to focus on is that there is no gap between one base pair and the next. Okay, so if we just rotate that round, you can see that very clearly there that here's a base pair, here's the next one, and they're stacked on top of one another with no gap in between. And that's hugely significant, because what it means is that the core of the double helix excludes the water that's surrounding this molecule in the cell. So out here, all around the outsides, there's going to be water, as we discussed when we were thinking about where the phosphate groups would go. But in the middle, where those bases are stacked on top of one another, there's no space, and the water's going to be completely excluded. And what we need to think about now is what's the significance of that in terms of forming a stable structure. So if we look at our base pairs again, one thing that we really need to take account of is that not all of the atoms that could potentially be involved in hydrogen bonding are actually involved in the base pairing. So we've got this set of 3 hydrogen bonds down here, but you can see, for example, in the case of cytosine, there's another NH there, which could be involved in hydrogen bonding, but isn't involved in the base pairing. On the guanine, again, you've got the nitrogen there, and you've got this hydrogen here, and you've got that nitrogen there. In this base pair, we've got the oxygen down here, nitrogen there, this NH up there. So there are lots of atoms which could be involved in hydrogen bonding, which are not directly involved in the base pairing. So what are they doing in the double helical structure? Well, if we look at the structure again, we can see the answer to that, which is that some of those nitrogens, which are blue, and oxygens, which are red, which belong to the bases, are actually not hydrogen bonding to one another within the base pairs, but are actually poking outwards. In other words, they're poking out into the solvent water. And you can see that from their point of view, that's not a problem, because water can form hydrogen bonds. And so what's happening is that this nitrogen and this oxygen, all these bits of each base which are not involved in the base pairing hydrogen bonds, are simply hydrogen bonding to water instead. And that should make us think, because what we have to do is think, well, what happens if it didn't form the base pairs at all? What would happen with the hydrogen bonding then?
So what we have to think about is, if this is our double helix with the base pairs in the middle, with the red dots represent those hydrogen bonds, that is basically going to be in equilibrium with an unstructured state in which those two chains are separate. So there's the purple one and there's the brown one, and they're now no longer wrapped around one another, and the bases will just stick out into the solution in each case. So obviously what that entails is breaking those hydrogen bonds. But as we've seen already from the groups within the base pairs which are not directly involved in base pairing, that actually isn't a problem, because all they do is hydrogen bond to water instead. So what we're going to do is get a bunch of water molecules hydrogen bonding to these bases instead of the bases hydrogen bonding to one another. And so effectively what we're doing is just replacing like with like. We're just replacing one set of hydrogen bonds with a different set of hydrogen bonds. So to a rough approximation, we can say that hydrogen bonding makes no difference to the stability, because all you're doing is replacing one set of hydrogen bonds with a different set. And so to a rough approximation, and obviously it is a rough approximation because we're kind of here assuming that all hydrogen bonds are equal, which is probably not realistic, but it's a reasonable approximation to say that the hydrogen bonding makes little contribution to the stability, because when it goes between a structured and an unstructured state, you just replace one set of hydrogen bonds with a different set of hydrogen bonds. So what that implies is that what makes the double helix stable is not the hydrogen bonding. On the other hand, what we saw when we looked at the space-filling model is that the core of that structure has no space for water. So when we drag some of those bases out of the water and force them into the core, if they didn't form hydrogen bonds, we would be in trouble, because they wouldn't be able to hydrogen bond to water because there's no water there. They have to have complementary hydrogen bonds. So now we begin to see why hydrogen bonding is important. It's not that it makes the double helix stable, because if the helix unfolded, they'd just hydrogen bond to water. They wouldn't care. But if it's going to fold up for some other reason, you have to force the hydrogen bonding in the middle to be correct. Otherwise, it's going to destabilize the whole structure.
Okay, so you can probably understand this better with the help of a soup-based analogy. So here is a bowl of delicious soup, and I have a strong driving force to eat this soup because it's nutritious and delicious, as you can see. However, this might happen. Now suddenly, even though I still like soup just as much as I did before, I feel disinclined to eat the rest of this soup because it's now liberally mixed with Labrador saliva. So the point here is that the driving force for eating soup is still there. The driving force isn't that I like soup because it isn't mixed with Labrador saliva. That's not what the driving force is. That's just a necessary condition that has to be met. The driving force is that I like soup, but unless the other necessary condition is met, which is that it's not mixed with Labrador saliva, then it's still not going to happen. And that's exactly the case with the DNA, that it will fold up, and the driving force is not the formation of those hydrogen bonds in the core. The driving force is something else which we've still got to discover, but unless that condition is met, unless those hydrogen bonds are formed, then it's not going to happen. And then you can have the rest of the soup.
So when you read in textbooks, as you will, particularly in A-level textbooks, but even actually at degree level, that the DNA double helix is held together by hydrogen bonds, you'll know now that that's kind of misleading, that really the hydrogen bonds don't hold it together. Something else must be holding it together. But that doesn't mean the hydrogen bonds are unimportant. They have to be correctly matched so that you can form those correct base pairs in the middle. And that can only really be done with the classic AT and GC base pairs, because anything else would destroy the regularity of the helix and therefore wreck whatever other interactions there are which are in fact holding it together. So that's a real important change in understanding from what you tend to get in most books when you think about the way DNA really works. But of course what we haven't talked about is what really is holding the structure together. And we're not going to go into that in huge amount of detail today, because obviously time is getting on and you're probably losing the will to live at this point. But we'll just talk about it a little bit. And the key is, once again, to go back to that space-filling picture that we had like that. And the point that makes is just how densely packed those atoms are together in the middle of the structure. And that is important for two reasons. The first is that, again, if you've studied some chemistry, you know about other kinds of intermolecular forces, not just hydrogen bonds. And in particular, you know about induced dipole-dipole forces, which are forces that can exist between any atoms, regardless of the overall polarity of bonds or anything like that. Any atoms can form induced dipole-dipole forces. But again, what's not really emphasized at A-level, but what's really important, is that those forces are extremely short range. So they only work as an effective means of sticking atoms together if those atoms can get very close together. So that tight packing in the middle of the structure is very suggestive of an important role for induced dipole-dipole forces, because that's the way it works. And because the bases are flat, you can see that each of them has a big flat surface which can pack against the flat surface of the neighbouring base pair. And the induced dipole-dipole forces between one flat surface and the next add up to quite a lot. And those undoubtedly make an important contribution to the stability, because when the DNA unfolds, if the two strands came apart and those bases were exposed to water instead of packed against each other, they would have far fewer induced dipole-dipole forces. Because the thing about water is that its structure is dominated by its hydrogen bonds. So the water molecules don't actually pack together as closely as they possibly can. They pack together in a way that optimizes their hydrogen bonding. So if those bases are exposed to water, they have fewer contacts with the water surrounding them than they would do with neighbouring bases there. So the induced dipole-dipole forces almost certainly play an important role in making this structure stable. But there is something else, and this is the last sort of idea that we're going to introduce today, because it's actually incredibly important in not just understanding the folding of DNA, but actually understanding the folding of other structures, especially protein structures. And that's what's called the hydrophobic effect. So we're just going to talk a little bit about the hydrophobic effect, which is probably the single biggest driving force for the double helix. Now, if we're going to understand the hydrophobic effect, we have to understand a little bit about the structure of water. So this is showing you what a water molecule does in a crystal of ice. So you can see that here's the water molecule, the red is the oxygen and the white are the two hydrogens. And you can see each water molecule, this one's just an example, forms 4 hydrogen bonds. So each hydrogen is forming a hydrogen bond. So this one, you can see it's that linear arrangement between the oxygen it's bonded to covalently. There's the hydrogen, and then there's this linear hydrogen bond connecting it to another oxygen. Same for this one up here. So each hydrogen forms 1 bond, and the oxygen forms 2 hydrogen bonds, each one directed at one of its lone pairs. So you've got OHO there in a straight line and OHO there in a straight line. And that will be true for every water molecule in the crystal, other than obviously those ones right at the surface, which are not going to have a full set of partners. But for the overwhelming majority of water molecules in an ice crystal, you've got 4 hydrogen bonds. Now this is a simulation of liquid water. And what you can see this time is that the water molecules individually have quite a lot of freedom of motion this time. So they're not in such fixed positions as they are in ice. But what you can also see is that they're still forming a lot of hydrogen bonds. So if we focus on this water molecule here, you can see at the moment it's forming a hydrogen bond to that oxygen, one to this hydrogen, and one to that hydrogen. And now another hydrogen bond has appeared there. So for the part of the time, it's forming its full set of 4 hydrogen bonds, but for some of the time it's forming fewer as the molecules move around. But despite that motion, the molecules are still very closely clustered together, and they're not sort of dashing around all over the place. They're just sort of oscillating around most of the time within a fairly limited area. Over a longer period, there will be larger scale migrations, but largely we're talking about hydrogen bonds being made and broken as time passes, but within a very limited volume of space. And within that volume, you can see that on a time average, they're forming typically around 3 hydrogen bonds. So not as many as in ice, but still a lot. And as a consequence, they're still very tightly packed together. So what we need to do is think about what the consequences are going to be for that dynamic structure of water if we try to dissolve something else in it, and in particular, something that's not polar. So here's another little simulation. So here we've got a bunch of water molecules in the liquid state. So you can see they're not organised in a tremendously regular way as they would be in ice, but as we saw a minute ago, they're quite densely packed together with lots of hydrogen bonds between them. And then up here, we have a non-polar molecule of some kind. And what we're going to think about is what happens if this dissolves in that cluster of water down here. So there it's gone into the heart of that cluster. And what's fairly obvious is that in order to make room for it in there, we're going to have had to move the water molecules apart from one another. In effect, we've had to make a cavity in the hydrogen bonded structure of the water because the non-polar molecule can't make hydrogen bonds. So the water molecules have had to prise themselves apart from one another, and all the non-polar molecule can offer in return are much weaker intermolecular forces, induced dipole-dipole forces, which are a very poor substitute for the hydrogen bonds that are broken. So that's a very unfavourable thing, which is why non-polar substances have low solubility in water. And we can see that that is going to be very relevant to what happens when the DNA folds up.
So if we think about our folding equilibrium, so here's a sort of crude representation of the double helical structure, showing now the base pairs with this kind of space-filling format, so that we can see that there are no spaces in between them. And then this is a sort of impression of what it would look like if this unfolded and the two strands were separate once again. And remember that the whole thing is bathed in water. So what's going to happen in this state is that there's going to be lots of water molecules all around the outside of the structure, hydrogen bonding to the phosphate groups and to the sugars and to those bits of the bases which are not directly involved in the base pairing hydrogen bonds. But there is going to be no water at all inside that core because there's just no space for it. But once it unfolds, the water's going to be surrounding everything. Okay, not just the backbone, which is still exposed, but all these bases as well. And that's the big difference, that you're now going to have water having to cope with these big flat surfaces of the bases. And although the bases have got all these polar groups around the fringes, they're flat structures, so the top surfaces, those are in effect non-polar. And so you can see that there's a huge difference in the solvation, because the water here, and in this molecule exactly the same, is having to cope with these non-polar surfaces being exposed. And as we've seen, that has a disruptive effect on the hydrogen bonding in the water and is an unfavourable thing. So when the two strands come together and squeeze water out from the core, then in effect these waters and these waters and these ones and so on will be stripped away, because these surfaces are now being packed against one another, and those waters will be returned to the bulk solution. They're no longer involved in solvating the bases, so they'll be squeezed out to here, and that's going to be much more favourable, because then they can get back to being involved in the hydrogen bonded clusters. And so the net effect is that it's favourable for the DNA to fold up, partly because you can get favourable interactions between the bases, and we talked about the induced dipole-dipole forces, but actually mainly because it's more favourable for the water, and the water can form a better network of hydrogen bonds when it doesn't have to cope with these big non-polar surfaces being intruded into its structure. And that's what the hydrophobic effect is, this idea that non-polar surface tends to get excluded from the water and pack against other non-polar surfaces, and that's what defines these structures.
There is a final twist in this tale, which is that when you look at the hydrophobic effect in more detail, what you find rather surprisingly is that the energy of the system when the non-polar surfaces are exposed to the water doesn't actually appear to be much higher. In other words, there doesn't appear to be a very big energy cost associated with breaking up the normal hydrogen bonded structure of the water. But instead, there is an entropy cost. So entropy, remember we said at the beginning, is this measure of how dispersed the energy actually is. And the fact that there's an entropy cost suggests that when the non-polar surfaces are exposed, what actually happens is that the water molecules become more organized around the non-polar surfaces. And you can sort of see that in this simulation, that the water molecules surrounding the grey blob in the middle, which is a non-polar solute, they lose that freedom to rotate. They're wiggling around a bit, but they lose that freedom to rotate and bond to different partners, which they had in bulk water. And that, of course, is because they can't form hydrogen bonds in the direction of where the non-polar solute is. So rather than accepting that they're going to form fewer hydrogen bonds, they reorganize themselves into a rather ordered layer around the non-polar solute. And that has the effect that actually the total number of hydrogen bonds doesn't change all that much. But the cost comes in the fact that those water molecules have to be more organized. And as a result, there's less rotation, so the energy is less dispersed than it would be if the water was just normal bulk water without that hydrophobic surface to deal with. So it brings us rather neatly back to where we started and this idea of entropy. So when we really pick apart how this beautiful double helical structure folds, or more particularly why it folds, what we discover is that the simple idea that it's hydrogen bonding between the bases is really not right. That's very important for stopping it from being unstable. But what makes it stable is the hydrophobic effect, and that is associated mainly with the entropy of the water surrounding the structure. That it has higher entropy, and therefore the energy is more dispersed when you tuck the non-polar surface away, because that non-polar surface has this curious ordering effect on the water. And losing that is what drives the double helix to fold. So it's a very interesting tale where it's easy to sort of focus on the part of the structure that's most evident, which is the specific base pairing, and assume that that's what makes it work. But actually, it's much more complicated than that. So we'll stop there for today, but there is a huge amount more you can learn about DNA folding and protein folding indeed, which is an even more complicated topic if you're interested in that.